I just want to start by saying this is a highly collaborative project with both alumni, so faculty, alumni, and current undergraduate students at the University of Tampa, as well as interns and faculty at Cape Eleuthera Institute. So it's been a great project in terms of getting lots of people and the community themselves involved. So the first thing I want to address is why seahorses in the Bahamas. We know that there are 3 species that are here. I've been described as being here, actually. And the other thing that's important to know about this group is that they are listed under Appendix II of CITES, and they are also, the entire genus is listed on the IUCN Red List, and the species that was identified as the ones in the pond is actually listed as vulnerable on the IUCN Red List. So there's conservation concerns about this particular species. Away? All right. The second thing that's important to know about this particular group is that seahorses worldwide are in a precipitous decline, and the Bahamas is in a unique position to offer baseline data on seahorses throughout the Bahamian archipelago. Because seahorse populations are under threat due to poor fishing practices like trawling, and the Bahamas has no tradition of trawling. It's target fishing here. And so as a result, this baseline population opportunity is a very exciting one in terms of seahorse biologists worldwide. The next component, of course, is that they've been used as indicator species for habitat quality in different projects around the world. So again, another reason why seahorses in the Bahamas should be of focus. And so this project actually started many years ago, back in the early 2000s, with me looking throughout the Bahamas for seahorses, and through Ethan Freed and Mark Daniels and colleagues at CEI back in 2013, I found out about the population in Sweetings Pond, and then the rest, of course, is history in terms of this project. So Sweetings Pond, Emily already introduced you to. This is a pond system that is actually quite large. It's 1 mile long by 1 mile wide, rather deep for these kinds of saltwater ponds, over 40 feet. And it has some unique coastline, here that separates it from the Caribbean side. The tidal change in the pond versus outside is only 10 centimeters relative to 1 meter offshore. And as a result, and there's no boiling hole in the pond. So the pond appears to be separated from the outside environment except for water flow changes. We don't believe there's actually species exchange between the outside ocean and the pond itself. So, previous work, Emily mentioned, was Aronson's work in the early 1980s. And Aronson's work established that this pond has low fish diversity. So his work established that there were only 17 species from 15 families inside the pond. And our follow-up work on that indicated that through our underwater surveys, in the intervening 30 years, we're only seeing 13 species from 12 families. So it looks like there might have been some attrition in terms of species in the pond since that time. Of course, Emily already talked about this being a predator-free system. And so Aronson's work with both the brittle star and the octopus indicated that their behavior and their physiology indicated that there were no predators, no natural predators of these organisms in the pond, which for us is interesting from the seahorse perspective, being no natural predators for them as well. Come on. All right, I'm going to do it this way. The last thing that Aronson's work established, which was good for us, is that being a PhD thesis out of Harvard, it was an extensive environmental survey. He also surveyed Benthic Habitat. So Aronson's work established that most of the bottom, the warm colors here in this bathymetry map, was covered by Cladophora algae, an algae associated typically with, it's a fast-growing algae that its growth is spurred by nutrients in the water column. Around the pond edge back in the 80s, there was much more agriculture at that point, and we believe that the growth of this algae that overgrew all of the benthic organisms that were there, killed off large areas back in his work, as he described, was kind of an important time point back then for us then to compare the work that we're doing now. So we've been able to do some comparative work on this project. Back then, he observed that seahorses were only seen less than 1 per dive, and that is not the case these days. We see lots more than that. So the community has changed in the intervening 30 years. So this leads to my research questions. There are 2. The first is, what does the benthic cover? So what's on the bottom of this lake or this pond? And does it match seahorse preference? Are they using habitat unequally relative to its distribution? And we're predicting that with more stable bottom community in the intervening 30 years with less agriculture on the pond edge now, that we should see from that point forward a higher density of seahorses that were seen initially by Aronson. In addition, we want to know how do seahorse population densities compared to those that we know around the world. And so we'll talk about that as well as we go forward. How we did this, Emily talked about a little bit of background work here. We took standard environmental variables, including variables such as nitrates and phosphates that are indicative of heavy nutrient loads, so we're measuring those as well. And we had 4 main phases to the research. We had 2 spring measurements, so March of 2014 and this 2016. So we just were analyzing data as of last Friday. And then 2 summer time points as well to have kind of a seasonal perspective on this. We ran 30-meter transects that were either 1 or 2-meter belt transects, depending on particular contexts, from 5 locations around the lake. These locations varied a lot visually in terms of their bottom habitat. We had one that was located, it's the site of an organic farm. It has gated access. This is Queens Highway along here. We have our second site, Quarry. Our third site, Grouper Cave. These make up our southern sites here. Quarry is a site where we're seeing an increase in ecotourism in the lake, so this is a site that humans can actually access. Control and Grouper Cave form our areas that are very hard for humans to get to. They'd have to kayak or swim a half a mile to get to these locations. And so these are kind of our baseline populations for any impacts of humans along the way. And then the Hatchet Bay Caves end of the lake, which is probably the highest impacted area from humans. At each location, we ran between 5 and 8 transects in those repeated 4 time points. On each transect, we took 30 photos, 1 per meter, to estimate, and we used those photos to estimate what the bottom habitat looked like across a number of major categories. So these are the major categories listed here, just to give you a sense of the species diversity we see on the bottom of this lake. And we actually specially focused on the algae that we found on the bottom of the lake because that appeared to be the most important habitat for seahorses in terms of where we found them. We used a program known as coral point count to randomize points to assess the percent benthic cover of all of these different components. Then animals, as Emily described, were photographed for in situ, so where we found them to identify where they were in the habitat, as well as photographs of their morphology to be able to assess reproductive condition, gender, all of those factors. So just from an environmental perspective, I'm going to go quickly through this one. This is a fully marine system, so it is salty. It's not brackish. It's a fully marine system. And we have seen, although our nitrates, and phosphates were low at the beginning of the project, it appears that some of these numbers are beginning to creep up a little bit, associated with a steady increase back to agriculture around the pond's edge. So what did we see in terms of bottom habitat? Well, we saw a range of habitats that varied again across the pond. Our first site, Emma's, was dominated by, so the coloration here, so this is the key that is listed roughly in abundance. So the most abundance categories across the board typically were algae across our sites, followed by flocculent and sediment. These are open habitats. There's no cover there. They're just one's sand, one's flocculent. And what you can see here in terms of Emma's site is that it is mostly algae, almost 50% algae, and the other 50% is open habitat. So it's just simply sand or open bottom. The numbers here are associated with the number of different categories that are represented in these pie charts to give you just a rough estimate of diversity of bottom that we identified in this location. So our 2 southern sites, Grouper Cave and Quarry, were particularly dominated by open habitat. So they have this flocculent habitat here, or open sediment. So not much algal cover in those 2 locations. Then in our other sites, so our 2 northern sites, Control and Caves, we have in these locations, these are dominated by algae. So the blue parts of those pie charts are all algal cover, followed by sediment, so open habitat, and then also some of that flocculent cover. In terms of these blue bands, so the next figure focuses in, this next figure focuses in on these blue bands just to look at what algal species we saw. And in terms of the algae, we saw two main categories of algae. Caulerpas are in this dark red kind of salmon color, and the Laurencias are in this light pink, and then also green was just a different form that we saw. So we can see of the algae we saw at Emma's site, most of it was either Caulerpa or Laurencia, with a few other species represented. What we saw at our two southern sites, remember these were the sites that had the lowest algal cover. These were sites that had less than a quarter of the site had algae covering the bottom. And in these locations we saw either Caulerpa in quarry site or Hypnea, which was one that we really only saw in high levels here and then some more up at caves. And then finally our last two sites, our northern sites, which were our algal-dominated sites, these were dominated by Laurencia, so it's mostly all Laurencia at our control site. And here we see mostly Laurencia with some acetabularia and other filamentous algae and some Hypnea here. The key issue I want you to pull away from this figure is comparing this back to Aronson's original work. We see that Aronson's work described the bottom is covered by that Cladophora algae. Cladophora is this bright green coloration, and you can see that in all of our sites there's very little of this algae relative to other algal species. So what were the seahorses doing? What were they holding on to at these different habitats? Well, what we see in terms of seahorse habitat use was if Laurencia was present, that is predominantly where these animals were associated. This is a brown, knobby algae, offers a lot of habitat, a lot of places for these ambush predators to be able to feed. Our two southern sites had very little algal cover, remember? And when there was less algal cover, the algae kind of diversified. You didn't get one dominating those ecosystems. And so the seahorses kind of diversified there where you would find them. And our two algal-dominated sites, you can see Laurencia. So when seahorses were present, we were seeing them most associated with this brown algae. So what were seahorse densities in this system? Well, they were high. So this line I'm going to put up here is worldwide densities. If we take all the seahorse studies and look at them, seahorses worldwide, Sweetings Pond numbers are much, much, much higher than we see in other populations around the world. We did see a seasonal effect in terms of their densities. The blue bars are our summer sampling. We see many more fish out on our transects in the summer than we see in the winter. We don't yet know where they go, but that's part of the next steps that we're looking at here. We also saw a north versus south bias in the lake. Fewer fish were seen on transects in the south relative to the north, probably because of the lower algal cover in those locations. Finally, looking at sex ratio, these are monogamous beasts. They've been found to be genetically monogamous, so we would expect a monogamous system to see roughly equal males and females. And what we see in terms of our seasonal effects, in the summer we tend to see more males on the transects. In the winter, we tend to see more females on the transect. So there is some bias in their sex ratio in this particular system. Again, something that we're looking to track. We're using actually mark recapture techniques now to be able to investigate these kind of patterns so that we can actually track individuals over time to kind of figure out where they're going and what it is they're doing. In terms of our conclusions, the first thing is that we saw great variation in benthic cover across the lake, and seahorses were preferring algal holdfasts and specifically preferring Laurencia where we saw it. And their densities are high compared to other seahorse populations worldwide. So this provides a very unique living laboratory opportunity and is an incredible, from my perspective, I've been working on seahorses my whole career. This is kind of the crown jewel of seahorse biology. There's a lot of seahorse biologists out there that are very excited about this system. The other thing that's important to remember, though, is that we did see changes in the bottom habitat. So early on, we had that Cladophora associated with we think, higher levels of agriculture. And so as agriculture on the pond edge moves forward, we need to be paying attention to making sure that not many nutrients make it into the water column, in addition to controlling sediment load to make sure that visibility isn't affected. So we need to be careful about and manage the upland areas to help protect this resource. And there is a lot of ongoing work in the pond. I just want to use this slide to say, please go see Katie Lynn Roberts' poster on Thursday. She's actually been working on a plankton study looking at food availability for these animals, and we'll be excited to talk about that work then. And also, we are working with the International Conservation Organization Project Seahorse to try to map seahorse populations throughout the Bahamas. And so if you are seeing them as biologists, there's an online app you can go report those, and then I can actually download that data and move forward to be able to get this information. And we have a lot of other research going on. We already discussed the concern about the marina and rising ecotourism and agriculture. So as we move forward from conservation perspectives, we need to be thinking about these issues and trying to manage the system very carefully as we go forward. So and I'm going to skip these last two because Emily already talked about them, which is awesome, and mention that the marker capture work is ongoing, and then say thank you to my collaborators, my funders, and then my students. And the other folks, this has been a strongly collaborative project with, of course, folks at the Leon Levy Preserve, the Bahamas National Trust, and then a host of undergraduates and interns that we've been working to train in this system. And I'll take questions.